Hello dear students, this is Dr. R. Haritha, lecturer in Biotechnology from Government Degree College for Men, Srikakulam. And today I'll be dealing with the topic monoclonal antibodies, also known as MABs, and the Hybridoma technology. That is the technique which is used to produce these MABs. So what are the learning objectives of this class? So by the end of this class, you'll have a clear picture about the introduction of monoclonal antibodies, and how to differentiate between polyclonal and monoclonal antibodies, and also study the principle of hybridoma technology, the various steps that are involved in this hybridoma technology for the production of MABs, and the applications of monoclonal antibodies. So what are actually these MABs or monoclonal antibodies? So any antibody that is specific against a single epitope that is present on an antigen is called as a monoclonal antibody. So you have that in your name. Mono is single and clonal means they're produced from one single parental B cell. So they're all clones of a single parental B cell that is highly specific to one single epitope on an antigen. So why am I stressing the point that it is specific to a single epitope? The reason is, if you remember, your antigens tend to have multiple epitopes on their surface. So one single antigen, several epitopes on the surface. And these monoclonal antibodies are so specific that they recognize only one of the epitope that is present on that particular antigen. So these are identical immune cells that are all clones of a unique parental cell. So that's what I was saying, monoclonal antibodies. So here you can see in this diagram. So this is the antigen, an antigen that we know of, any antigen you can think of. And you have, in this case, 4 different epitopes. So one single antigen having several different epitopes. And these monoclonal antibodies are specific to only one of that antigen. So usually what happens when an antigen having multiple epitopes enters into our body, we produce polyclonal antibodies. So antibodies that are specific to each of these epitopes. So those are known as polyclonal antibodies. Whereas monoclonal are specific to only one single epitope that is present on the antigen. So what are monoclonal antibodies? Antibodies with the same specificity produced by a cell line that is derived from only one activated B cell and which are able to recognize only one epitope. So that is what we were saying. Monoclonal, single, mono is one. So you can say in terms of one single epitope that is being recognized, and clonal means they're all produced from a single B cell. One single activated B cell is the parental cell of all the monoclonal antibodies. So here you can see you have antigen with several different epitopes on its surface, but our monoclonal antibodies, which are being depicted in blue, can recognize only one single epitope, one specific type of epitope of all the different types of epitope that are present on this specific antigen. Whereas coming to polyclonal antibodies, these are a mixture of antibodies with different specificities. So they can recognize the epitopes, the different epitopes that are present on a single antigen, and also their parental cell is not one single B cell, but they are different clones or different cell lines of B cells. So they recognize different epitopes on an antigen. So monoclonal antibodies refer to homogeneous population of antibodies that are produced from a single clone of plasma B cell. Whereas polyclonal refers to a mixture of antibodies that are secreted against one single antigen. So here in polyclonal it is single antigen, whereas in monoclonal it is single epitope. The specificity is further enhanced in monoclonal antibodies. So monoclonal antibodies, all the monoclonal antibodies will be produced from a single plasma B cell, whereas polyclonal will be produced from different clones of plasma B cell. And also monoclonal can interact only with one single epitope on the antigen, whereas polyclonal will interact with different epitopes on the same antigen. And monoclonal antibodies production, when it is done in vitro, that is in lab conditions, it's highly expensive, whereas polyclonal antibodies is not that expensive as compared to monoclonal antibody production. Yeah. And usual uses of monoclonal antibodies are usually used for as therapeutic drugs. We will see further on in this class how they can be used as drugs, whereas polyclonal antibodies are generally used for usual basic research applications. So advantage of monoclonal antibodies are that they are immortal. The cell lines of monoclonal antibodies are immortal. So we have an unlimited supply. They're also highly specific and high reproducibility. So every time you produce them, that specificity will be retained. Whereas with polyclonal antibodies, the advantages is that they have higher affinity, they have tolerance to minor changes in the structure of an epitope, whereas monoclonal cannot bind if even a minor or a single bond change occurs in the structure of an epitope. And also polyclonal have the advantage of being more robust detection. So the conditions need not be as sensitive or as controlled as when we are using monoclonal antibodies. So each set of antibodies have their own advantages. So coming to the hybridoma technique or the hybridoma technology. So these monoclonal antibodies are produced using a technique which is known as hybridoma technology. And this technique was developed by two scientists. You can see them here. You have Georges J.F. Kohler and Cesar Milston. And for this discovery, the development of this technique, they have been awarded the Nobel Prize in 1984. So this hybridoma technique is a technique that is used to develop monoclonal antibodies that are specific to a single epitope from a hybrid cell that can grow continuously. So what is this? We will see in the next slides. So coming to a normal B lymphocyte. So what is the characteristic of a B lymphocyte is that it can produce antibodies that are specific against a single epitope. But the problem is that these normal B lymphocytes are motile cells. That is, they divide up to a number of times and then they tend to apoptose and die. But we need an unlimited supply of these monoclonal antibodies. So what did these two scientists devise? So they have identified that we need to immortalize the B cells. So whatever the B cells that are producing the antibodies that we require, these B cells have to be immortalized. That is, they have to become continuously dividing cell lines. They should not die. So what is the technique that they have developed? So what they did is they have selected a myeloma cell or a cancerous cell. So the basic characteristic of a cancerous cell is its immortality. It does not die. Even after unlimited cell divisions, it still continues dividing. Whereas normal cells are all motile cells. Usually after 50 times of cell division, our cells tend to apoptize. So they have devised this hybridoma technology on the principle of the fusion of an immortal myeloma cell and an antibody producing plasma cell. So the basic idea is that the plasma cell has the characteristic to liberate or secrete your antibody, and the myeloma cell has the immortality characteristic. So they have fused these two cells to give rise to a hybrid cell, which is known as hybridoma cell. So ideally, this hybridoma cell or hybrid cell should have characteristics of both the parental cells. So from plasma cell, it gets the characteristic of antibody production, and from the myeloma cell, it gets the characteristic of immortality. So our hybridoma cell will have both the antibody producing capacity and the immortality. So how this is what we have just explained. So the target is that we need an immortal B cell that can synthesize the antibody of our choice. So how do we get this done? This is achieved by fusion of a somatic cell hybridization between a normal B cell that produces the antibody and a myeloma cell. So this fusion gives rise to your immortalized antibody producing cell lines, and these are called as hybridomas. And the antibodies that are produced from these hybridomas are called as monoclonal antibodies. So what are the characteristics of this hybridoma cell? So as we were just saying, this hybridoma cell has two basic characteristics. One, ability to secrete antibodies, and this character has been given by the B lymphocyte. And the second characteristic is unlimited cell division or immortality, and this is given by your myeloma cell. So hybridoma cells are hybrid cells that will continuously produce one specific type of antibody. Simple as that. So moving on to the steps in hybridoma technology. So how is this fusion achieved and how do we actually select the hybridoma cells? That is what we will be seeing now, and this entire technique was developed by the two scientists Kohler and Milstein. So the first step in hybridoma technology is immunization of the mice. So whatever antibody that we need, the monoclonal antibody that is specific to one single epitope, that antigen is introduced into the mice. So this is known as immunization of mice with that specific antigen. So once the mice is immunized with that antigen, what happens? The B cells that are present in the spleen of that mice, that is the B lymphocytes, will get activated and they will start producing the specific antibodies. These are known as sensitized antibodies, activated antibodies. So next step is what? The fusion between a myeloma cell and a B cell, the B lymphocytes that have been sensitized. So any fusion that occurs, for example, we are fusing 100 B lymphocytes and 100 myeloma cells, we cannot guarantee that all the B cells and myeloma cells will be fused together. So after the fusion, we will have a mixture of cells. We'll have unfused cells. We'll have the same type of cells that might fused with each other, and we'll have the hybrid cells. From all this mixture of cells, we have to select the specifically hybrid cell that we need. So for that selection step, we will now introduce a small modified type of cells. So what is this modification that we are talking about? So the myeloma cells, in the first step that are fused with this B lymphocytes have been modified such that they cannot produce an enzyme. This is hypoxanthine guanine phosphoribosal transferase enzyme, HGPRT. H hypoxanthine, G for guanine, P for phospho, R for ribosal, T for transferase. So when I say that it is HGPRT minus, it means that particular myeloma cell lines cannot produce this enzyme, right? And the second type of parental cell that has to be fused is the spleen cell, which has this activated B cells. So these spleen cells, they have not been modified in any way. So these are HGPRT plus, that is the gene, the HGPRT enzyme producing gene is active in the spleen cells. So these spleen cells are completely normal. They have all the enzyme producing genes and they are motile and they secrete the specific type of antibody because that mice has been immunized with our antigen. The myeloma cells, the cancer cells which are immortal, these are the ones which have been modified such that one single enzyme which is known as HGPRT, this enzyme producing gene is switched off. Okay. So what is the significance of this HGPRT enzyme that we are talking about? So remember, this HGPRT modification has been introduced for the selection step. So it has no role in fusion. This will play an important role or a crucial role when we come to the selection step. So what is the role of this HGPRT enzyme in the biochemistry? So coming to the nucleotide synthesis. The nucleotide synthesis in mammals occurs in two pathways. You have a classical pathway, you have an alternate pathway. And another interesting fact, this is just an additional fact, is that most of the biologically important pathways have two ways. You have the classical pathway and you have the alternate pathway. Any problem if encountered in the classical pathway and that classical pathway is shut down, then that biomolecules will be synthesized using the alternate pathway. So nucleotides we know are very crucial biomolecules that are required for any cell to survive, right? So nucleotides in the sense they're the building blocks of your nucleic acids, DNA, RNA. So without DNA and RNA, there is no life, right? So in nucleotide synthesis, the de novo pathway, the pathway that you can see here, this pathway is the classical pathway. So usually when everything goes well, so the synthesis of nucleotide occurs using the de novo pathway. Whereas when we come to the other pathway mentioned here, which is the salvage pathway, this is the alternate pathway. So whenever any problem is encountered in de novo pathway and it gets obstructed or switched off, then nucleotide synthesis occurs using the salvage pathway. Salvage pathway, you again have two different starting materials and two different paths. So one is using thymidine as a starting material, and the most important key regulating enzyme here is thymidine kinase. Whereas the other pathway is the hypoxanthine precursor using pathway, and here you have this HGPRT enzyme as a key regulating enzyme of this pathway. Hypoxanthine guanine phosphoribosyl transferase. So now you remember that the myeloma cells that we have used for the fusion are HGPRT minus, and we'll come back to this later on. Now, and the fusion. How does this fusion occur? So here you can see the yellow colored B cells, the yellow colored B cells, which are HGPRT plus, that is the enzyme is active. The gene that is synthesizing the enzyme is active, whereas the myeloma cells depicted in red are HGPRT minus. So now we have to fuse these two cells, and the fusion is occurred using fusogenic agent. And this fusogenic agent might be biological or chemical. Biological in terms of inactivated Sendai virus can be used for the fusion of both these cells, or when coming to the chemical fusogen, the most commonly used fusogen is PEG, that is polyethylene glycol. So during this fusion process, you have two steps in which the fusion occurs. So these fusogens, in this case, for example, polyethylene glycol, will bring both the cells that have to be fused, that is the normal B cell and the cancerous myeloma cells together. And at first what happens is the plasma membrane fuses and the cytoplasm of both the cells become fused. But the nuclei as such are separate, intact, and they are separate. So at this stage, this cell is known as heterokaryon. In the name again, hetero is different, karyon is nucleus. So you have two different nucleus in the same cell because the cell membrane and the cytoplasm of the two cells, that is the B cell and the myeloma cells, are fused, but the nucleus has not yet fused. So this is the heterokaryon stage. In the second step, what happens? Both these heterokaryons or both the nuclei will fuse together and to form a single cell with a single nuclei. At this stage, it becomes the hybrid cell or the hybridoma cell. So you can see in the picture, you have the blue cell, which is the T cell, the B cell, and the red colored cell is your myeloma cell, and these both are fused. At the first step, you have the heterokaryon formation. You can clearly see the two nuclei, the blue and the red, separate. And at the second step, you have the hybridoma being formed, which has a fused nuclei, a single nuclei containing the genes of both the B cells and the myeloma cell. So as I was saying earlier, so for example, if we have fused 100 B cells and 100 myeloma cells, it is not guaranteed that all the 100 B cells will go and fuse with your 100 myeloma cells. You have a mixture of cells at the end of the fusion step. So you have unfused cells, that is B cells as such might not fuse with any other cell. Similarly, you have the myeloma cells which remain unfused. And then the second type of cells that are found are the fused cells. In the fused cells, again, you have two B cells that have fused together, two myeloma cells that have fused together, and the third type of fused cells are a B cell and a myeloma fusion. So this is what our target hybridoma cell is, a B cell fusing with a myeloma cell. Excuse me. But as I have mentioned, we have four other different types of cells in the mixture. Unfused B cells, unfused myeloma cells, B cell fused with a B cell, myeloma cell fused with a myeloma cell. So from this mixture of cells, we have to select our hybridoma cell. And for this selection step, which is the next step, the third step, we use a specialized medium which is known as HAT medium. H-A-T, HAT medium. So the full form of this is H for hypoxanthine, A for aminopterin, T for thymidine. So this is what HAT medium is. So what happens in this medium? So for the nucleotide synthesis, we had the classical pathway which is known as the de novo pathway. This de novo pathway is blocked by the aminopterin that is present in the media. So the aminopterin that is present in the media will block your de novo pathway of all the cells. Okay. And coming to your aminopterin is over hypoxanthine. I told you hypoxanthine is the starting material for the salvage pathway, the alternative pathway. So if we have hypoxanthine and if the enzyme HGPRT is present, then this pathway will go ahead and will result in the synthesis of nucleotides. So what happens to the different types of cells that we have found or we are left with at the end of fusion? We'll come to unfused B cells. So unfused B cells are HGPRT plus. That is, the enzyme is present in those cells. So what happens in those cells? Usually de novo pathway will be obstructed because aminopterin is present in the media, but they can synthesize nucleotides using the salvage pathway as they have the HGPRT enzyme in them. But we know that B cells are motile cell lines. That is, they die after a specific number of cell divisions. So even though initially these unfused B cells can survive on the HAT medium, they do not have the immortality component, so they die after few cell lines. Now come to the unfused myeloma cells. Myeloma cells, we have modified them so that they do not have the HGPRT enzyme. So what happens? De novo pathway is blocked because of the presence of aminopterin, and salvage pathway cannot go ahead because they do not have the HGPRT enzyme. Hypoxanthine is provided in the medium, but the enzyme is not present. So myeloma cells unfused myeloma cells cannot survive in your HAT medium. So the same is the case with B cell fusing with a B cell and a myeloma fusing with a myeloma. They have the same characteristics. B cell has the enzyme HGPRT but cannot survive for long because of its motility character. Whereas immortal myeloma cells, they do not have the HGPRT enzyme, so they do not survive in the medium. So what are we left with? We are left with hybridoma cells which have the HGPRT enzyme from your B cells and the immortality character from your myeloma cells. So of all the 5 types of cells that we were discussing earlier, only your hybridoma cells can survive in a HAT media. So in the slide you can see you have the blue colored B cells and the red colored myeloma cells. So unfused B cells will lead to senescence or cell death because they are motile cell lines. A B cell fusing with a B cell, that is 2 blue cell lines fusing, will also have only motility component and they will die after few divisions. Coming to your myeloma cell, the red colored single cell, unfused myeloma cell also dies because aminopterin blocks your de novo pathway and HGPRT enzyme is absent in these myeloma cells. coming to fused myeloma cells. Two myeloma cells fusing with each other. Two red cells. Again, they have the immortality component, but they cannot survive because there is no HGPRT enzyme in them. So the only option that we are left are a blue colored B cell fusing with your red colored HGPRT cell. So these hybrid cells or hybridoma cells as they are known are the only ones which will survive in your HAT media, because even though de novo pathway is blocked, the salvage pathway will be active because of the HGPRT enzyme that is present in the B cell that has been fused with your myeloma cell. And myeloma cell will give the characteristic of immortality. So they can divide for any number of cell divisions. So no senescence. So this is how the selection step occurs. Right. So and another sub-step of the selection is that we have to select the actual clone that will produce your antibodies of that are specific to one particular epitope. Because remember, I had showed you, let me go back a little and I'll show you what, yeah. During fusion, you can see here that after heterokaryon formation and during hybridoma formation, you have some chromosomal loss that we have depicted as random chromosomal loss. So that is not under our control. And when the two nuclei are fusing with each other, randomly some genes will be lost. So sometimes what happens? Some of the genes that are lost might be the genes that are of our interest. They might be the actual antibody secreting genes. So that is the reason we have this selection of clone choice also that has to be considered. So from antibody secreting hybridomas, the first thing, pure clones must be screened for the desired antibody specificity. So whatever the antibody specificity that we are aiming for, that specificity should be present in the antibodies that are being secreted by these hybridoma cells. So that is what we call as clonal choice. So some clones will be there which will produce antibodies that have unwanted specificities. That is, that specific to some other epitope rather than the epitope of choice that we want. So this differentiation or removal of these unwanted clones can be done using your ELISA and RIA techniques. So ELISA and RIA techniques are used for screening which clone from these hybridomas will be producing the desired antibody that has the specificity to the epitope that we want. Right, and come the final step, that is the propagation of the desired clone. So from selection again occurs at two steps, remember. First step is from all the other types of unfused and other fused cells, we have to select the hybridoma cell. That is done using the HAT media. And the second type of selection that we are talking about is clonal selection. So those clones have to be selected which will produce the antibody of the desired specificity. So once that cloning is done, now we move on to the next step, that is the propagation. So we have to propagate or multiply our desired clone that we have chosen. So this can be done in vitro and in vivo. When we are talking about in vitro, it is done using tissue culture. So these MABs are cultured for indefinite period because they have the immortality characteristic in a culture medium. But the disadvantage of this technique is that we have very low concentration of antibodies that are secreted into the medium from the cells. So the low concentration, we are talking in terms of 10 to 100 micrograms per ml. There is a second step, second type of propagation, that is in vivo, in mice. So these hybridoma cells are propagated in the peritoneal cavity of the mice. Remember, the histocompatibility of the mice has to be matched. Otherwise, we will have the rejection problem. So the histocompatible mice, in those, the peritoneal cavity is the place wherein these hybridoma cells will be propagated. And by this technique, we have a higher concentration of antibodies that can be obtained, the monoclonal antibodies. So this concentration is around 1 to 25 micrograms per ml. So tissue culture or in vitro, we are talking about micrograms, and whereas this is milligrams. So you can see the difference. So this is the overall pathway of the hybridoma technique that we were discussing until now. So you have an antigen that has multiple epitopes. So you have the naming and the different coloring and the different structural representation also. So this particular antigen has around 4 epitopes. When this antigen is introduced into the mice, in under normal conditions, what happened? We get polyclonal antibodies. So antibodies that are specific to the different epitopes that are present on that antigen. So you have antibody 1, 2, 3 and 4. Correspondingly, they're specific to that specific epitope. But when we are talking about monoclonal antibodies, so what we do, we isolate the spleen cells containing the sensitized B cells that are active against one specific epitope of all the 4. So you have the 1, 2, 3, 4. So each B cell that is active against that specific epitope. And then we have, they are fused with the myeloma cells. We get these hybridomas. And these hybridomas are again, clonal selection is done, and the different clones, all the hybridomas that are monoclonal, capable of producing monoclonal antibodies that are specific to epitope number 1 will be collected in one test tube. Here you can see the second class or the line of clones are specific to the second epitope. So each monoclonal antibody's clones are specific to one particular epitope that is present on a single antigen. That is the level of specificity that we are talking when we talk about your monoclonal antibodies. So what are the applications of these monoclonal antibodies? So the most common application is the in vitro diagnosis. So any diagnosis when we are talking about systemic or infectious diseases that are based on detection of a particular antigen or in fact an antibody also, these are done using your monoclonal antibodies in the different types of immunoassays. You like what we have seen, ELISA, RIA, all these use monoclonal antibodies for detection. And regular screening, clinical diagnosis also, like blood grouping test, pregnancy test, or measuring the various levels of drug usage test, all these are also, for these detections also we use monoclonal antibodies that are specific to the epitopes that are present on the target cells. Coming to in vivo diagnosis. So until now we have seen in vitro, in the lab, the diagnostics that can be carried out using monoclonal antibodies. Now we will talk about in vivo diagnosis. So what we can do is we use radio labeled or fluorescent tagged maps. So we tag these monoclonal antibodies with our fluorescent tags or radioisotope tags, and then these are introduced into the body to identify where that particular antigen is present in our body. Best example when we are talking about tumor antigens. So MABs are usually very commonly used, to detect the presence of breast cancer. So these MABs are labeled with radioisotope iodine 131, and these isotope labeled MABs are introduced into the patient's body. These are highly specific to the breast cancer, cell surface receptor. So wherever the breast cancer is there, or you can also, it is just not for the presence or absence of breast cancer, you can also determine the spread of the tumor, to what extent or to how much the tumor has spread, the tumor growth has spread, can also be detected using your MABs. Coming to uses of monoclonal antibodies in therapy. So it can be used to identify your major histocompatibility gene or the different microorganisms for clinical diagnosis, as we have already discussed earlier. And one major use of these monoclonal antibodies is they are used as immunotoxins. So what are these immunotoxins? Immunotoxins, in terms of they're used as delivery vehicles. It's just not for diagnosis, but they can be used also for treatment. See, here you can see you have three common types of bacterial toxins. You have ricin, you have Shigella toxin, and you have diphtheria toxin. These toxins, they have one or more binding chains. So what we do is, one of the main, the toxic chain is binded to a monoclonal antibody. And this monoclonal antibody is specific to the cell surface receptor on a target tumor cell. So what happens? So when this immunotoxin that is specific to the cell surface receptor on a particular tumor cell is introduced into a patient, this will act as a delivery vehicle and it will take the toxin chain and deliver it directly to the specific target cell. So for all the cells that are present in our body, only that particular tumor cell, to that particular tumor cell, your immunotoxin will go and attached on the surface and introduce or liberate the toxin into that particular cell. So this is how or monoclonal antibodies can be used for treatment also. So you can see what happens in normal conditions when a diphtheриal toxin comes and binds to the cell surface receptor is endocytosis. It is taken inside and endosome is formed, and from this the toxin is released and it will inhibit the protein synthesis because it inhibits the elongation factor that is used in the elongation step of translation. And when we come to the immunotoxin, the mechanism is exactly the same. These immunotoxins are the monoclonal antibodies that you can see here. They're highly specific to the cell surface receptor that is present on tumor cell. So tumor specific antigen. So this antigen is the target of your immunotoxin. So your immunotoxin comes and binds to that tumor cell. The same process, endocytosis occurs and endosome is formed, and the toxin chain that has been attached to your MAB, monoclonal antibody, is released, and this will inhibit the protein synthesis in a tumor cell. So what happens? Ultimately, the tumor cell dies. So the mechanism of action is same, but instead of a toxin being delivered to a healthy cell and a healthy cell getting killed, here what we are doing is only the target infected or the tumor or defective cells, the target cells are killed. So this is targeted treatment. So you have another application of monoclonal antibodies where they can be used as enzymes. So you combine an antibody with an enzyme, it becomes abzymes or CatMab, catalytic antibody. So what do they do? The same procedure. These enzymes can be used to cut peptide bonds at specific amino acid residues. So within the protein, if you want any specific modifications wherein a specific bond between two particular specific amino acids has to be broken, that specific peptide bond can be broken. This exactly acts like restriction endonucleases that are used, if you remember, to cut DNA at highly specific sites. They have a recognition site, a specific sequence, and only in that sequence specific cuts will be made so that the surrounding genes are not disturbed. Exactly like this, these abzymes can be used to make specific cuts within a protein structure so that they can be used for structural and functional analysis of proteins. So these were the different applications of MABs or monoclonal antibodies. So in this class, we have studied actually what are monoclonal antibodies and how are they different from polyclonal antibodies, and what is the procedure, what is the principle of their production using hybridoma technology and the various steps in hybridoma technology. And finally, we have ended with the applications, the various and many applications of monoclonal antibodies. So I hope this class was clear enough to you. Thank you.